In this video, we're going to take a look at antibacterial drugs, otherwise known as antibiotics. And so we'll take a look at the structure as well as how they act on bacteria in order for them to do the job that they need to do. Now, really interesting to note that these are one of the most frequently prescribed classes of drugs out there, and they are toxic to bacteria but relatively safe to humans. And so they're going to act on sites and bacterial cells that are either different from human cells or do not exist in human cells. There are many different types. The one we're going to focus on in this video are the penicillins. So interesting about the discovery of penicillin, and you can go take a look and see what other videos you can find on the discovery of penicillin. But it was actually discovered by chance in 1928 by Alexander Fleming. And basically, it was found to be produced by some fungi of the Penicillium strain, known as Penicillium. I don't even want to say that word, so you can try and say it in your head. I'm not good with the Latin terminology here. But essentially, it was discovered by chance, which is kind of really interesting. In terms of the structure of penicillin, we'll look at the general structure first. So it's a bicyclic structure, and that's because cyclic means rings. So there are 2 rings within the structure. It contains a beta-lactam ring, which is a cyclical amide that is part of a 4-member ring. So when we're talking about this beta-lactam ring, we're talking about this 4-membered ring here. It is essential for antibacterial activity, and we'll see why momentarily. But that's a really, really sort of tough ring to have there. It's a good idea to pause the video here and just take a look. What other functional groups are present? Where are the chiral carbons in this molecule? So things like we've got a group here, a group here, a group here, actually that together, and this thing. So go through and name what those functional groups are and see if you can find any chiral carbons. All right, so let's move on, and we're going to talk about penicillin G specifically, or benzyl penicillin. It's one of the most important natural penicillins because it was the first one to be isolated and purified. Essentially, how they make it in these big vats, and they manufacture it by fermentation of a mixture of corn steep liquor, sugar, minerals, and this phenylethanoic acid, using a penicillium fungi in a very carefully controlled environment. And so it creates this penicillin G. The G stands for this side chain here, and we'll talk about side chains in a minute, but basically it's a phenyl group with another carbon methyl group attached to it. Now, one of the major disadvantages of this particular penicillin is that it's easily broken down by stomach acid and has to be given by injection, which is not exactly very easy to do. And we know that when we take penicillin nowadays, we do not take it by injection. So like I said, with penicillin G, we actually have what's called a side chain here. And in order to create the kind of penicillins that we take today, we can actually modify this side chain to create ones that are more stable and that we can take orally. So modern or semi-synthetic penicillins, so things like ampicillin, are penicillin molecules where the side chain has been altered to modify or alter its properties as well. And like I said, advantages are it reduces the occurrence of penicillin-resistant bacteria. And it also is resistant or more resistant to the breakdown by stomach acid, so we can now take it orally. There are lots of different side chains that are available for different types of penicillins. Now, how penicillins work, we first kind of need to talk a little bit about bacterial cell walls. So, bacterial cell walls are really interesting in terms of their structure. They have these sugar chains that are cross linked with peptides via an enzyme called a trans. Trans just means across, and then peptidase means peptides or proteins, essentially. And so we create this sort of structure here. Now, this gives strength to the cell wall. It can withstand really, really high osmotic pressures, so that's great for the bacteria. Penicillin, though, comes along, and the way that it works is it acts on the bacterial cell walls. So it's going to work by deactivating the transpeptidase enzyme. Essentially, it becomes an irreversible inhibitor. And what happens there is the bacteria needs to, essentially, it's presenting the formation of the crosslinks within the cell wall itself. And so, as a result, the walls are going to be weakened. The bacterial cell is going to absorb too much water, so lots of water is going to come rushing in, and it's going to burst as a result of an increased osmotic pressure. So let's take a look a little bit closer. Like, how does this react with the transpeptidase? Well, it comes back to that beta-lactam ring, which is essential to the mode of action of penicillin. So penicillin's going to come, and it's going to come and bind into the active site of the transpeptidase. So this green box is just depicting the active site of that enzyme. And in the active site of that enzyme is this OH group. And it's going to act as a nucleophile. It's going to attack the carbonyl, which is then going to break open this carbon-nitrogen bond. So it becomes attached then to the active site, and this is irreversible. So we're creating a covalent bond between the enzyme active site and the penicillin, leading to the deactivation of that transpeptidase enzyme. Now, bacterial resistance can form, and so resistant bacteria produce an enzyme called penicillinase, which is an enzyme that's going to break open the beta-lactam ring in the penicillin molecule. Now, I did forget to mention this beta-lactam ring is very, very reactive because the bond angles are very strained there. And so it wants to break open, essentially, because those bond angles, it's really tough to hold it within that sort of ring structure. So it's very susceptible to nucleophilic attack. And so resistance is going to arise because of mutations in the DNA of bacteria to aid their survival. And that resistant bacteria is going to reproduce and pass on their resistance to successive generations. And so bacteria, get passed down with this penicillinase. And the more that we expose bacteria to antibacterials, the more opportunities there are for the bacteria to mutate into bacteria that have genes that are going to produce this penicillinase. So it's really important to only take penicillin when we absolutely have a bacterial infection that requires us to get rid of it. And so that comes along to sort of our last point here is patient compliance. It's very, very important that antibacterials are taken according to doctor's instructions and that the whole course of treatment's taken. So not stopping in the middle of a treatment. And that's because if you fail to kill all the bacteria in the infection, that can lead to further resistance of those bacterias and that do survive. And so it becomes harder to then kill if you need to take another round of antibiotics. So really, really important that you comply when you are taking antibacterial medications. So we've looked at the structure of penicillin, we've looked at how it works, and what leads to bacterial resistance. That's it then for this video. We'll see you in the next one.